Hello everyone, welcome back to Dr. K. Primer lecture series presenting by Dr. K. That's me. Today we will talk about expression vector, most used expression vector for recombinant proteins. That is none other than PET21A. So before going to the PET21, let us talk about the categories of vectors. Vectors are two categories. One is cloning vectors. The second category is expression vector. Cloning vectors are used to amplify the cloned fragment in a more copies. For example, PBR322, PUC, PUC18 are cloning vectors. Then coming to the expression vector, which are having the elements which are necessary for high level of transcription and translation of cloned gene. Expression vector carries the elements which are required for high level of gene expression and followed by the translation of cloned gene fragment. Such vectors are called as expression vector. So when you clone your gene of interest into the expression vector, you can express high transcripts and high protein or more protein. So elements which are required for the strong expression means high transcription means strong promoter. So if you clone your gene of interest to the downstream of under the control of strong promoter, that will synthesize the more transcript. Once we have more transcripts, then that will turn into the protein and more protein polypeptides you can get. So expression vectors are used to express the protein of your interest, right? So those things we will talk. Those what are the features of clone expression vector? We'll discuss in this lecture. Yeah, PET21 is the most popular expression vector, and PET21 is having more variants like PET21A, PET22B, C, D. That all depends on the open printing frame. I'm here going to present the general features of PET21 expression vector. So this is the backbone of PET21, whose size is 5,443 base pairs. And as I told you that expression vectors have elements or sequences which are required for the expression, strong expression of a clone gene. So one of them is strong promoter from T7 bacteriophage. This promoter is isolated from the T7 bacteriophage and inserted into the, kept in this vector, under which there are different elements which are required for the transcription and translation. The first one is RBS. So when you clone your gene of interest under the T7 promoter, and T7 promoter facilitate the transcription of your gene, which will consist of RBS, ribosome binding site. Ribosome binding site is required for the initiation of the translation of your transcript. Whenever transcript is coming from the DNA, which is if it is having a RBS, ribosome binding site, there ribosome complex is binds, then initiate the translation of transcript into the polypeptide. So sometimes some genes may have, may not have, but RBS also there, inbuilt RBS is there in the vector. And also there is a leader peptide or T7 tag, after the RBS. T7 tag, that's 11 amino acid length T7 tag of G10 of T7 bacteriophage. This T7 tag helps in the detection and quantification of a recombinant protein. So T7 tag which is present at the N terminus of N terminus, which will be there in the cloned recombinant one, and which helps in the detection as well as the quantification of recombinant proteins. After T7 tag, there is a polylinker site or multiple cloning site where a series of restriction sites will be there. By using them, you can clone your gene of interest. So polycloning site, polylinker or MCS, multiple cloning site, and the PET21 have BAMH1, ECOR1, SAC1, IND31, EGG1, NOT1, ZO1, avian. These are the restriction sites which are there in the multiple cloning sites of PET21. So you can use any two of these restriction sites to clone your gene of interest. That's what MCCS. And after the MCCS, there is a one more element sequence. That sequence is called as histidine tag. Histidine tag. So histidine tag will be adding to your cloned gene fragment, and it is translated to the 6 histidine amino acids in your protein. So histidine tag relevance is that whenever you want to purify your recombinant protein from the E. coli. E. coli is having around 4,000 proteins, right? So out of which you want to purify your only protein, right? So the histidine tag will help you to purify single step purification. So when you pass the recombinant E. coli lysate through the column chromatography, which is having the nickel column, so the recombinant protein with the histidine tag will struck to the nickel, and rest of the protein goes through the flow through. So histidine tag will help you to purify your protein, right? So there are, again I'm saying, T7 promoter for a strong transcription, RBS for ribosome binding site and initiate the translation, T7 tag, which is required for the detection and quantification of recombinant protein, and MCCS for cloning of your gene of interest, and histidine tags will help you to purify the recombinant proteins from the complex or lysate of the E. coli. So these are the elements which are required for the transcription and translation of the cloned gene fragment. And there is a strong T7 terminator also, because sometimes some genes carries stop codon, sometimes they don't carry their own. So if there is a T7 terminator, then the transcription stops, then the transcript undergoes for translation, right? So this is what is the main difference between the cloning vector and expression vector. And also there is one more interesting feature in the PET21 is F1 origin. F1 origin. So this F1 origin, whenever you transform the PET21 into E. coli, and if you infect that with the helper page F1, then the helper page will synthesize the single strand DNA of clonal fragment from using this F1 region. So why do you require the single strand DNA molecule? Because you want to sequence the cloned DNA fragment. So you can use the alpha page to synthesize the single strand from this construct. And one more interesting one is ampicillin resistance marker, which is required for the required for the selection of transformants. Whenever this vector is transformed to the E. coli, which is sensitive to the ampicillin, the transformed E. coli are become resistant to the ampicillin, and they grow in the presence of ampicillin. That is provided by the ampicillin resistance marker or gene called as beta lactamase. So ampicillin resistance, beta lactamase is useful in selection of transformants. And there is one more feature, that is the origin of replication required for the autonomous replication of plasmid. And there is one more element which is also important for the transcription of the clone gene, that is LAC-I. That's a repressor. LAC-I is isolated from the LAC operon, where this encodes the repressor molecule. That repressor molecule binds to the operator region in the promoter and stops the transcription. So, in the absence of, this is useful in the regulation of gene expression. Whenever you want switch on the gene expression, then whenever you want to shut down, on and off is depends on the, you can regulate through the LAC-I repressor, right? So these are the features of pET21, and I hope you understand. And the same thing I'm going to explain with the next slide also. So T7 pET21 is having a T7 strong promoter. Why? For high expression of clone gene, strong transcription. And T7 tag, that's a 11 amino acid peptide encoded in the letter sequence of T7 bacteriophage gene 10, will help in the detection and quantification of recombinant proteins. Ribosome binding sites required for initiation of translation. Multiple cloning site with 8 restriction site facilitate the cloning of variety of DNA fragments. Tag, that is a 6 histidine amino acid for single step purification of recombinant protein through the nickel affinity column chromatography. Strong T7 terminator stops the transcription of clone gene. F1 origin for synthesis of single strand DNA of clone fragment for DNA sequencing. Ampicillin resistance marker or gene that is encoded by the beta lactamase gene, which breaks down the ampicillin into penicillinic acid and provides the resistance to the transferment. Original replication for independent replication of plasmid, and this is a low copy number of plasmid pET21, which provides the stability and sustainability to the recombinant plasmid in the host. And IPTG is the important molecule, as I said. IPTG will induce the transcription of clone gene, or in the presence of that, in the absence of IPTG, there is no transcription of your clone gene, right? So this is what is the pET21 expression vector, and hope you enjoy my lecture. If at all, then you please subscribe my channel. That's a Dr. K PREM-PRIMER. And if you like it, and try to subscribe, and if you more like it, try to share with your friends. And if you have any doubts, please write to me through the comment section. Okay then. So see you all again with one more video. Until then, bye bye.